The final talk today will be given by Gurmeet Singh. He's a professor and center head at the Disciplinary University right here in Bangalore in India. This will be on the theme of natural products and how their chemistry intersects with the chemistry of the human body. Gurmeet.
Thanks, Mukund. Hello, everyone. We've heard about the future of chemistry, and I think what I'll do is now go to the past and see what are the insights that we can get from the past and how they can be relevant to tackle today's challenges. So I'm going to talk on cross-cultural perspectives, focusing on food chemistry. There's a growing realization that chemistry and biology need to work more closely together, right? There have been, if you look at last 2 decades, there have been a flurry of papers and books with titles such as The Need for Chemistry and Biology Interface. A few journals have been launched over the last 2 decades as well around. But in fact, in nature, chemistry and biology are not distinct, right? They are studied as one. And in fact, holistic approaches of studying nature are rooted in the 2 sciences being studied as one. One such knowledge system is Ayurveda, right? It studies the chemistry of ingredients and their processing in the context of human biology in a field called Dravya Guna. And through its systematic epistemology, Ayurveda has characterized more than 1,500 plant species besides minerals and animal origin products as well for their effect on human body, right? And it uses a multi-scale framework to do so. That includes defining ingredients in terms of their 5 phases, the space, gaseous, fire, liquid, solid, and then characterizing them by their in-mouth experience called rasa in Sanskrit: sweet, salt, sour, bitter, astringent, and pungent. And these in-mouth experiences lead to 11 pairs of biophysical properties, how these materials would behave within the human body, whether they'll be light or heavy, they'll be heating or cooling, they'll be dry or unctuous, smooth or rough, thinning or thickening. And this leads to their potency and their post-digestive metabolic effect, which eventually builds up to the effect on what is called as effect on doshas or the bioregulation. And finally, to their specific therapeutic activities. There are about these 1,500 ingredients have been used to formulate about more than 4 lakh biologically functional formulations that have been curated in the traditional knowledge digital library available online by the Indian government's CSR, the Council of Scientific and Industrial Research. Right. So let's take the example, right, to build this forward, of a specific group of plant ingredients whose biology of taste is characterized by astringency, right? The Kashaya group, as it is called in Ayurveda. A number of the superstars of Ayurveda actually belong to this group of plants. Plants like Embelice officinalis, which is the Indian gooseberry, Terminalia cebula, Vitis vinifera, which is known to everybody as grapes, right? And Punica grantum, which is pomegranate, and many more, right? And some of these are actually becoming stars of the supplement world as well. Resveratrol from grapes and ellagic acid from pomegranate, right? But these astringent group plants are all rich in a certain group, in one group of phytochemicals called as phenolics, right? And the interesting thing about phenolics is that this is the most widespread and abundant group in our diet, and therefore it has generated huge interest from a human health perspective over the last 2 decades or so. However, this group is also one of the most confounding group of phytochemicals for scientists. For chemists, it has been a difficult journey to actually separate them, purify them, separate them to be able to understand their structure. There's so many of them in every plant, so many different molecules in every plant. And they oxidize very easily. They react with proteins. They react with carbohydrates. They react with the metal ions present in food, and it becomes very difficult to characterize them. Right? For the biologists, they are equally difficult because they don't seem to be very bioavailable. And there are papers after papers trying to understand how they could be effective if they are not very bioavailable. Right? So yet, if you look at the number of Ayurveda formulations using this group of plants, that suggests that there is a strong biological role there is. Right, and this is on this chart. I'm just taking a partial mapping of Embelice officinalis, the Indian gooseberry, and its benefits in the benefits of its various formulations, showing about 34 different health benefits. Put the Sanskrit names on there because the relationship between the Sanskrit names, the Ayurvedic disease descriptions, and Indian sometimes is not one-to-one. But maybe if I just risk it, some of the top ones are fever, skin disease, diabetes, cough, rejuvenation, eye disease, and anemia. So let's just take a few examples from this list. No, and on my first slide, I talk about the role of Ayurvedic formulations, especially amla-based, Embelice officinalis-based formulations in iron uptake. Now, iron deficiency, anemia, probably about 20 to 30% of the world suffers from it. And in India, if I look at the numbers, at the time of pregnancy, nearly half the pregnant women, 50%, are anemic at some point, which really affects the health of the infant as well. And so we've been studying the uptake of iron in a model system, globally accepted as a good replica of what would happen in a clinical study, and that's the CACO2 cell monolayer. And you take a digest of a food and you feed it to this monolayer and see how much of iron is taken up by the cells, and then move beyond and into ferritin, etc. And what we see is that there is a increase in iron uptake when, for example, we've been studying green leafy vegetables and how iron would get absorbed from them. And we do see that there is a significant increase when we feed these to the cells with and without, in the presence of Embelice officinalis, right? Literature does show similar things, and in many such publications, it's been pointed out that this could be coming from the presence of ascorbic acid in the Indian gooseberry. But we've done experiments where we've also looked at ascorbic acid equivalent levels, and we've seen that the bump up that we get because of the Indian gooseberry is way more than we would get from an equivalent amount of Indian gooseberry. And especially if you, the data here, which shows that when you process the food, the vitamin C levels, the ascorbic acid levels go down. And so what we may be having here is a temperature resistant, something which is possible for processed foods, a way to improve their iron uptake from them. Another area where this has been the Embelice officinalis has been used is in the area of glycemic control, diabetes, right? And again, a number of papers have studied, we too are studying the effect on digestive enzyme inhibition, right? The alpha amylases, the alpha glucosidases. There's also publications that some of the other metabolic enzymes involved in the metabolic pathways, such as DPP4, etc., can get inhibited by this. So there is probably not just, while this chart is showing just one aspect, it may be playing molecules in the Indian gooseberry, may be playing a more systemic role, right, besides just this. A third interesting example is the use of the Indian gooseberry extracts to stabilize oils. The Ayurveda formulary use a number of formulations actually are botanical extracts extracted in butter oil, or ghee as it is called. And the one challenge when we try and do it just directly is that this makes the ghee extremely unstable. Ghee, any other oil, if you want to infuse, it becomes extremely unstable, prone to rancidity. So how does this all work? So if you read the literature, it shows that there is a pre-processing step which involves treating the ghee with the juice of the Indian gooseberry, which seems to stabilize oil. And when we do this study and look at the stability in a accelerated oxidation instrument, right, where we take the butter oil, you put it into a cell at 130 degrees centigrade. You blow 20 liters per minute of air in it and oxidize it very rapidly. Under these conditions, what we see that a control butter oil sample would start degrading at about 5 hours or so, which is what this line here shows. And if you add the general stabilizers, the BHAs, the THQs, it probably adds from 5 hours, it goes to about 5.5, 6 hours or so. But if we follow the method given in the classical text, the stability goes all the way up to at least 25 hours, not more, 130 degrees. In real terms, that would mean that a butter oil that is typically stable for about 6 months or so would go on and become stable for about 2 years or so. So that's quite significant. And all this studied from the impact of how to make formulations more effective and get rid of the acidity. So those are quite interesting results that we're looking at. Now, many of these actions probably can be explained. Some of these, at least, actions can be explained based on the chelation, metal ion chelation properties of polyphenols, the antioxidant properties arising from the structure of the polyphenols. And here I show some 4 or 5 very popular polyphenols, and each of them you can see have multiple hydroxyl groups. Which can deprotonate easily because of the stable resonance structures which are created, and therefore that contributes to the antioxidant properties. Or the bidentate groups, the orthodiphenyl groups, for example, which lead to their iron chelation properties, right? So understanding these structures do help explain some of these benefits. However, the scientific studies, the examples that I gave, and the structural explanations, though promising, have not fully unearthed, have not unearthed the full potential of these ingredients that comes through a reading of the Ayurveda. So there's fuller study requires new thinking, new scientific tools, is a synergistic transdisciplinary research approach to deconstruct these Ayurveda formulations and come up with insights that can lead to more contemporary solutions. Now, of course, these new methodologies are coming up. Surely last decade and a half has been revolutionary in that perspective. I talk about 2 of them very briefly, not that they are the most important, but just because I'm a little more familiar with those pieces of work. The first is about the polyphenols of black tea, a drink which is consumed by more than half the world 3 times every day. And they have a group of molecules called thiorubigens, which were described first in the 1940s, 50s by a chemist called Roberts, right? And the first one to actually start talking about polyphenols, and then by Eddie Haslam in the 70s and 80s. But no real separation of these has been possible. And whatever HPLC we do, we got what we call as the TR hump, the thiorubigen hump, with only a few spikes, right? And what is hidden underneath this hump has not been known. And this paper by Kuhnert, which appeared around 2013-14, was quite revolutionary in that sense, using mass spectrometry and along with some analytical tools that the computational tools that he proposed. He explained the whole reactive chemistry, how 6 polyphenol molecules, the catechins, go to nearly about 30,000 or so species in black tea, and those kind of approaches can really unlock from a chemistry point of view the whole polyphenols. Another more recent story which has been emerging and very exciting story is the mediation that polyphenols. The microbiome could be mediating the role of polyphenols, and therefore probably this whole unraveling this whole bioavailability question, right? The papers here highlight how the ellagic acid in many foods such as pomegranate and Indian gooseberry, etc., how our microbiome acts on it and converts it into a more available form, which eventually leads to urolithin, which has very interesting bioactivities such as mitophagy, and could be could work towards longevity and the rejuvenation that Ayurveda talks about. So these are very exciting pieces that are happening. But what we have to make sure that as we apply these new tools, that we do not, these new approaches, which can help us deconstruct some of these formulations and generate new insights. But we need to make sure that we don't lose the systemic approach or the systemic sense of the whole thing. Systemic to ensure that the insights coming from pairings of botanicals, right? It's not just one molecule that we are looking at, or even one plant extract. Why are there so many different plant extracts in a formulation? So why are these pairings there? Why do we use fresh ingredients in some and we use dry ingredients? The degree of oxidation, the pH, the role of pH or speciation. Why some of these require fermentation externally? Is it to do with the microbial transformations, the mode of delivery, the seasonal implications? There are so many perspectives that need to be brought in to get to the specificity which seems to be articulated in the classical text, and also the systemic approaches from the network pharmacology and the action on the multiple subcellular processes, that we talk about: the mitochondrial dysfunction, the autophagy, the membrane flexibility, free radical generation, insulin resistance, inflammation, epigenomics, etc. So I think just to sum it all up, last point, that we are poised today where we can truly expand our horizons by combining systemic and molecular perspectives. These perspectives are complementary, and when put together, they can generate deeper insights which could address the complexity and the personalization which many now feel is needed to find solutions to the health challenges that are facing us. Yeah. So thanks, and we'll be happy to look forward to the discussion that follows. Yeah.